So we are shifting gears from the pig genetics to the virus genetics, and it's appropriate because when you think about it, selecting a good attenuated virus vaccine from a field virus is really selective breeding of the virus. So I've been with Zoetis and legacy companies for about 24 years now. Almost every day I spend part of my time thinking about what makes a good attenuated virus vaccine, PRRS and other veterinary viruses, and how can we better select for and recognize when we have a desirable phenotype in a modified live virus. So a couple of things I want to go over today. Introduction to conventional modified live vaccines, and this is a PRRS conference, so we'll focus on PRRS. The roles of viral genetics in vaccine efficacy. Selecting the appropriate starting strain to make a vaccine, and a little bit about the attenuation process that I kind of think of as premeditated serendipity. That is, if you go in with open eyes and an open mind, and you are aware of natural selection, you can look for and find desirable phenotypes in the virus that you want to be your vaccine. This table is basically a list of all the commercially licensed modified live PRRS vaccines in the world. It's not necessarily comprehensive, but I think it's close. The ones in orange are Zoetis vaccines. One thing to note is that I think there are 18 different modified live vaccines that are registered in the world today. Could be more. In the U.S., we have FOSTERA PRRS. It was launched about 6 years ago. In Europe, this is news. For those of you in Europe, you'll be seeing Suvaxyn PRRS MLV appearing on the market next month in January. We got market authorization in September. In China, we sell something called Rulanan, which is a high-path PRRS vaccine. This one is interesting. It has an extra-large deletion in NSP2, and that happened spontaneously at passage 19 and correlates very well with attenuation. The rest of the world, some countries have their own homegrown licensed vaccines, Philippines, Japan, Singapore. And then the major vaccines that are available in the U.S. and in Europe are also registered in different countries. FOSTERA PRRS, for example, is now registered in Canada, Mexico, the Philippines, Malaysia, South Korea, Thailand, and most recently Japan. So what are the benefits of modified live vaccines? Well, the commercial vaccines that are modified lives are currently the best solution we have for controlling PRRS. They seem to be far more efficacious than killed vaccines or experimental subunit vaccines. They mimic natural infection, and we know that when you have homologous challenge, you get very robust cross-protection, sometimes even sterilizing immunity. So that's kind of our gold standard. We know it can work under the right conditions. Modified lives induce all branches of the immune system. Innate immunity, so type 1 interferons, natural killer cells, also acquired immunity in the form of T helper cells, cytotoxic T cells, and B cells for neutralizing antibody. Modified lives don't require an adjuvant, although sometimes it's added. They can be delivered intramuscularly, and you only need one shot to protect a feeder pig for its life. So we have customer convenience with these modified lives. So a lot of things go in to determine whether or not a vaccine is efficacious under a certain circumstance. Some of those are under control of the vaccine designer or the vaccine company. For example, selection of the parental virus strain determines the starting genetics of your vaccine. The attenuation method and the endpoint of the attenuation determines the final genetics of your vaccine virus. The company also controls the formulation of the vaccine and the label recommendations. So we negotiate with the regulatory authorities what's the appropriate storage, the size and number of doses, routes of administration, age of the animal at vaccination based on clinical data mostly. Many things aren't under control of the vaccine company. These are in the domain of the customer for the most part. The genetics, health status, and nutrition of vaccinated animals. The immunological history of a herd, and this is previous vaccinations, previous exposure to field strains, environmental conditions such as weather. The actual usage and storage of the vaccine. We can make recommendations, but the recommendations aren't always followed exactly for various reasons. Also, there's the genetics, the virulence, and the immunogenicity of the challenge virus or viruses themselves. We can never predict for sure which viruses the herd will be exposed to. And finally, co-infections of other pathogens, either during the vaccination phase when we're trying to establish immunity, or during the challenge when other pathogens may exacerbate disease. So how do we select a starting strain? Well, it's a very important first step, so we have to do it first. And we put some thought into what would make a good starting strain, keeping in mind that it can take 5 to 7 years from starting strain selection to licensed vaccine product. So does it make sense to use the most recent hot strain as the parent of a new vaccine? For example, 174 we're hearing about a lot now. Well, you could do that, but that's a risky strategy for a couple of reasons. These new variants come and go frequently. They don't always leave behind economically important descendants by the time you would register the vaccine years later. And the other thing is that, and this is not intuitive, it's not obvious, but the recent field strains have evolved so much from the common ancestor that they're not closely related to each other and wouldn't necessarily provide good cross protection against each other. Sometimes an older ancestral strain is actually a better choice if the goal is broad cross protection. And in order to illustrate that concept, I want to show you a couple of patterns of evolution. This is canine parvovirus. Why? Well, I don't know. It's a good example of a linear, stepwise evolution of a virus. CPV appeared in the late 70s from feline panleukopenia virus, and that was CPV2. It changed over time through specific amino acid mutations to become CPV2A, 2B, and then 2C. So that's an example of linear stepwise evolution. The ancestral strains may be replaced either completely or partly by the new strains. The overall diversity, genetic diversity of the virus population grows only slowly, and vaccine update decisions are relatively simple, theoretically, to think about. You have to run the appropriate studies. In contrast to that pattern, though, this is PRRS, right? So this is a slide from Mike Murtaugh. And on the left, you see European PRRS. Let's see if I can point to that. And on the right, North American PRRS. This is PRRS 1. This is PRRS 2, using the right nomenclature here. Within PRRS, you see a lot of different branches going in different directions from some common ancestors. The ones near the tips of the longest lines are most likely the most recent in time. If we look at a couple of them, for example, let's look at this one and this one. And what's the genetic distance between those two? Well, it's the length of this line segment and this one added together. So that's quite a genetic distance. If we were to use this as a starting strain for a vaccine, it wouldn't necessarily cross-protect well against this one, and vice versa. So sometimes an ancestral strain that would map somewhere in here shares more epitopes with both this one and this one, and would give the best broad cross-protection against at least this whole cluster, if not some of these as well. And that's really why you see that the vaccines that are out there are largely based on older strains. The characteristics of this is that the diversity increases exponentially with time, and that the vaccine update decisions are quite complex. This is more data illustrating the same point. These three in red are relatively recent field isolates from Europe, one from Belgium, one from Germany, one from Australia. These numbers in red on the table are the genetic identity, nucleotide identity, between the three recent isolates. The numbers are fairly low. The mean is 85%. The ones in, I guess that's green, are four of the European vaccines, commercial vaccines, including our new one. These 12 numbers are the genetic identities between the four vaccines and the three field strains. And the average here is about 87. It's not a big difference, but it's in the direction you might not intuitively think. The genetic similarity between the vaccines and the field strains is actually greater than the similarity between the different recent field strains. So again, this kind of illustrates that point. So another question that comes up a lot: does genetic similarity predict cross-protection when you think you know your challenge strain and you want to select a vaccine strain? And it's a complex issue. The answer is yes and no. So let's examine the arguments for yes. So obviously, acquired immunity works on epitopes. So you need some degree of genetic relatedness in order to get any cross-protection. If we look at the extremes, 100% identity, so this homologous challenge with exactly the same virus, you infect, you wait a month or two, you try to reinfect with the same virus, uniformly that protection is very strong. That's because you have 100% amino acid identity, and by definition you have 100% shared epitopes. If you back up a step and look at heterologous protection within type 1 or within type 2 PRRS, you get variable results with cross-protection, ranging from very good to pretty poor. But it's almost always detectable, right? So if you have a good control group and enough pigs, you can usually see a statistical benefit to using the vaccine, but your clinical signs are still there. They're just not as severe as they might otherwise be. If we look at the numbers, that's usually between 80 and 99% amino acid identity. 99% amino acid identity, though, if you do the math and assume an epitope is 10 amino acids long and assume that any change in those 10 makes it a different epitope, 99% amino acid identity is only 90% shared epitope, so you've lost 10%. If you do the numbers for 80% amino acid identity, you're down to only 11% of the epitopes are shared between the two viruses. So it's really not a surprise that efficacy drops off the further away you get. If we go back another step and look at protection between PRRS 1 and PRRS 2, again, usually you can detect protection and a vaccine benefit if you have enough pigs in a proper control group. But now we're talking about 60% amino acid identity, and only about 1% of the epitopes are going to be conserved between a type 1 and a type 2 PRRS virus. And if we take one more giant leap, if we look at two related viruses with a common ancestor, but they're not the same virus, for example, two nidoviruses, like PRRS, an arterivirus, and PED, a coronavirus, that both infect pigs. But I think we don't need to run the study. We know from the field there's negligible or undetectable cross-protection between them, even though they may have a couple of shared epitopes in the conserved regions. Let's look at the arguments for no. It's not a good comparison to make. And the best argument there is there's so many other factors at play besides genetic relatedness when considering one vaccine versus one challenge virus. It's just hard to predict. So the immunity of the first virus is based on its ability to replicate the high titer, so that's antigenic load, its ability to induce immunity, its immunogenicity, how much or how little it interferes with host functions, how much immune dysfunction that virus causes, also the health and genetics of the host animal, and dependence on no interfering co-infections. Later, during the second infection, the challenge, Things to consider are the challenge dose and timing, the inherent virulence of that challenge virus, whether that virus has immune evasion or immune dysfunction mechanisms at play, and which ones, the age, as well as the health and genetics of the host animal, and again, co-infection with other pathogens. So it's sometimes the case when the existing vaccines are more related to each other than a particular challenge virus. So this is one big clade, for example, lineage 5, 8, and 7 U.S. modified lives, and a lineage 1 challenge virus, which is in a different large cluster. The small differences between the degree of genetic relatedness are insignificant and are swamped out completely by these other factors. Right. Especially if you're dealing with OR5, to make your genetic decisions, that's only one small gene in a big genome. This is a poster we presented 2 years ago at this meeting. I've added the green trend line since then. But the idea is to look at what is the relationship between OR5 nucleotide divergence going from homologous to cross-type, or type 1 PRRS versus PRRS 2. And a measure of protection, this is % reduction of lung lesions. And when we look at this is 12 different studies with FOSTERA PRRS. And when we look at the studies that had the most related challenge virus, so this is about, 5% divergent, we get pretty good. We get 75 to 95% reduction in lung lesions. As we get further away, out here beyond 10%, though, I think the trend line is indicating that we still have protection, right? There's still some shared epitopes and so forth, but I think the degree of protection is gradually going down. We typically will get around 50%. This is actually a European PRRS challenge, and there was 50% reduction in lung lesions. It's notable what's not on the trend line. Down here, this is high-path Asian PRRS. A very virulent virus, I think, is kind of overwhelming this measure of protection. This is like a 30% reduction, but it's from like 90% down to 60%. And I think this may be an artifact of the way we're calculating % reduction and the fact that it's a very virulent virus. But I would say even in those studies, the vaccine made the difference between live pigs and dead pigs in this very virulent challenge. So the attenuation process itself, we start with the assumption that all field strains are efficacious. They're just not safe. So we fix that by passaging many times in the cell culture system, and this results in two different kinds of changes in the virus genome. The first is a very rapid selection for changes in the virus that confer fast growth in the new cell line, so adaptation to the new cell line. This often results indirectly in reduced growth in the natural cell type over time. The other kind of mutation is a gradual accumulation in those genes that aren't being used. And these include the genes that modulate the immune system, because there's no immune system in cell culture. So one of the first rules of evolution is use it or lose it. And if you're not using these genes, you begin to accumulate random mutations, deleterious mutations that may downregulate or alter the function of these defensive genes, viral defensive genes. So as this is going on, we periodically check for safety or attenuation of disease, and also for efficacy. And at different passage levels, there's three possible outcomes. You can have an under-attenuated virus, that is, it's still efficacious, but it's not yet safe, so we need to passage some more. It could be over-attenuated, that's very safe, but no longer efficacious, because it can't grow well in the pig. So it's been through too many passages. Or you can have something that's appropriately attenuated. That is, it's become safe, but it's still efficacious, and this is a vaccine candidate. And because we're looking at a random accumulation of mutations, you never quite predict how long it's going to take or what you're going to get. And we see all of these. Every time we do an attenuation, we see all three outcomes. The window of opportunity between safe and efficacious can be large, which is always nice. It can be small, which means we have a limited window in which to make a master seed and commercial product. It can be non-existent. It can even be negative. Negative means that by the time it's finally safe enough, it's no longer efficacious at all. So that's kind of illustrated in this picture. But it doesn't say it here, but this is kind of what we did for Suvaxyn PRRS in Europe. We started with a number of different field isolates that we thought were appropriate for parental vaccine. We tried to expand them once on PAMs. Some of them couldn't even be isolated on PAMs and fell out at that stage. If they grew on PAMs, the very next passage they went onto this baby hamster cell line that expresses porcine, CD163. This is what we use to attenuate and grow FOSTERA PRRS and Suvaxyn PRRS. And some adapted well, some didn't adapt. The ones that did, we can now do biological cloning. 3 rounds, so that from there on, each lineage is going to be based on a single genome sequence. And they're not all the same because we're starting with a quasi-species. And after a number of passages, we get ready to do an animal study. And that means scaling up and making some high-titer stock for vaccination. Some of the viruses didn't want to produce a high titer, and they dropped out at that last step there. Others went into the study. Some of them were unsafe. They were under-attenuated. Some were not efficacious. They were over-attenuated. And a couple of them were just right and went on to become vaccine candidates. And one of them went on to become the commercial vaccine. So I said multiple, because you get a different result every time, you want to do multiple parallel attenuations like I showed, because every attempt will give a different result. Having said that, though, if you pick your starting strain and your attenuation conditions, including your cell line correctly, and you're aware of the selective pressures involved, you can limit the spectrum of possible outcomes, and that can work in your favor. So, case in point, to the best of our knowledge, all commercially licensed live PRRS vaccines in the world, except 2, have been attenuated on the same cell line, and that's MA104, or derivatives of that cell line, like MARC145, for example. And since we discovered CD163 as a receptor, we now know why MA104 and derived cell lines are unique among commonly used cell lines in their ability to grow PRRS. Because of a rare mutation, that cell line expresses the CD163 protein, the monkey CD163, even though that protein is normally expressed primarily in alveolar macrophages, but generally in monocytes and macrophages. Okay. So what are the 2 exceptions? And it's the ones I mentioned. FOSTERA PRRS and Suvaxyn PRRS MOV. These were attenuated on that hamster cell line that expresses the porcine CD163. So here's kind of a table showing the 2 attenuation methods in use now. On the left is the monkey cell line approach. More than a dozen commercial vaccines use this. So you have a strong selective pressure at the beginning for the virus to adapt its proteins to recognize the monkey CD163 better, and therefore get a growth advantage at the beginning of the attenuation process. The amino acid sequences of simian monkey CD163 and porcine CD163 are significant, so they have to make some changes. And then, when you put that vaccine back into the pig during vaccination, the virus finds itself having strong selection again to adapt back to the porcine CD163 better. So you're forcing the virus through 2 selective events, one during attenuation, one after vaccination. And incidentally, so there may be a trend. Let's look at this. None of those vaccines have been able to get a claim for use in nursing pigs as young as 1 day of age. Don't know exactly why. It probably depends on the vaccine, maybe safety, efficacy, maybe both. But in contrast, on the method we use, We attenuate on cells that have the porcine CD163 engineered into them from a constitutive promoter. And they're permissive. You can adapt the virus to grow in those cells quite easily. The virus is not forced to change to recognize a different receptor, as far as CD163 is involved at least. So it doesn't have to undergo that kind of a change. And then when you put it back in the pig, again, it doesn't have to adapt and change the virus's genetics in order to grow well in the pig that just got vaccinated. So it tends to be a more stable attenuation phenotype. And both of these two vaccines that we've done this way have earned a label claim for use in pigs that are only 1 day old or any time after 1 day of age, because we were able to show that they were both safe and efficacious when administered at that age. And so, you know, one went through USDA, one went through EMA, and was able to get the identical claim. So let me finish up here with a couple of conclusion slides. For now, modified lives are the best solution we have for controlling PRRS. It's not really cost effective or necessary to develop a new vaccine for every new strain that comes along. It takes a lot of time, it takes a lot of money. Because of the radial pattern of PRRS evolution, maximum cross protection can sometimes be provided by using an ancestral strain that shares more epitopes with all the recent strain than they do with each other. So at a high-level view, there's a clear relationship between genetic homology of two viruses and their ability to cross-protect against each other. It's kind of obvious. This is a consequence of the shared or conserved antigenic epitopes. But at the ground level, in real life, using OR5 sequence to compare from a limited number of vaccines, the one that's going to match a particular field strain, is kind of an exercise in futility. The small differences between viruses, the vaccines that are not that different from each other, are swamped out by the many other factors that confound the genetic effect of cross protection. So we attenuate by passaging many times on a permissive cell line, leading to increased safety and manufacturability while trying to maintain immunogenicity. The attenuation process is generally stopped when the vaccine has become safe enough to pass the standard safety test. Additional cell passages are usually not done because you risk over attenuation if you keep going. So although we're looking at random accumulation of mutations during attenuation, and that's unpredictable, strong selection pressure, if you're aware of it, and they can imposed by different cell lines, can influence the spectrum of possible outcomes. And in some cases, you get this safety stop. And we think it may be, we don't have all the data yet, we think it may be a general function of monkey cell attenuation, that the virus, in the process of acquiring higher affinity for the monkey CD163, it loses affinity for the porcine CD163. And we know those vaccines tend to give a very low titer out of the bottle when titrated on primary macrophages. So that could give you a safety stop during attenuation sometime, maybe most of the times, where the vaccine becomes safe because it can no longer recognize the porcine CD163 very well. It's not all or nothing, but it's less efficient infection of macrophages. And that kind of a safety stop we don't get when we use porcine CD163. We keep going. We get attenuation based on other mechanisms. For example, I mentioned gradual mutations building up in the genes that modulate the immune system. So we have this alternative system for attenuation now. And it looks like they may have novel properties, beneficial properties in terms of safety and efficacy in pre-weaning pigs as young as 1 day of age. So I think my time's up. So I'm going to stop here, and thanks for your attention.